The targets of gene therapy are the genes carried on 23 chromosomes and the proteins they encode. Most of us have 46 chromosomes, a set of 23 from each parent. Successful gene therapy depends as much on manipulating cells, including immune cells, as the genes inside the cells. The germline, or the sex chromosome, that produces sperm or eggs is the 23rd in each sex. It's usually not targeted because the result of DNA manipulation can be passed to children. Genes tell cells how to make proteins that power enzymatic speeding of chemical reactions. That power the work of structural, storage, transport, hormonal, defense and receptor proteins. What is gene therapy? Many genetic diseases are caused by different genes working together: diabetes, hypertension, Parkinson's disease, peptic ulcers, pain, cancer, heart disease, autism, Alzheimer's, ADD, ADHD. Other genetic diseases are caused by a single gene: Huntington's disease, cystic fibrosis, sickle cell anemia, hemophilia, Huntington's disease, muscular dystrophy, bubble boy disease or severe combined immunodeficiency. There are more than 6,000 single gene disorders, occurring in about 1 in every 200 births. Gene therapy is a methodology used to introduce genetic material into cells to compensate for abnormal genes and chromosomes. Variations of genetic therapy. Germline gene therapy. Genetic manipulation of sperm or eggs, the germline chromosomes, can cause permanent change that can be passed on to future generations. The therapy can be effective as a permanent fix where family members have inherited a defective gene. The therapy is controversial because potentially defective genes can also be passed to children, and altered genes can be used just for cosmetic effect, a kind of eugenics. Somatic cell therapy. Therapeutic genes are transferred into the somatic cells of a patient. Modifications will be restricted to the individual patient, and will not be inherited by the patient's offspring or later generations. Ex vivo somatic gene therapy. Cells are modified outside the body then transplanted back. In some cases blood or bone marrow are removed and reinserted using a virus. Bone marrow ex vivo gene therapy was successfully used to treat bubble boy disease. In vivo somatic therapy, genes are changed in cells still in the body. It's been successfully used to treat HIV AIDS, Parkinson's disease, blindness, diabetes. How improved genes are delivered. If a mutated gene causes a protein to be faulty or missing, gene therapy can introduce a normal copy of the gene to restore the function of the protein. The normal gene is usually introduced into cells by a viral vector. Adenoviruses are viruses that carry their genetic material in the form of double-stranded DNA. They cause respiratory infections, the common cold, intestinal and eye infections. In humans, they introduce their DNA molecule into the host. The genetic material of the adenoviruses is not incorporated into the host cell's genetic material. The DNA molecule is left free in the nucleus of the host cell and not replicated when the cell divides. So descendants of that cell will not have the therapeutic gene. Herpes simplex viruses is particularly appropriate for delivering genes to neurons in view of its ability to establish latent infection in these cells. The virus can be used to treat such neurological conditions as malignant gliomas, Parkinson's disease, stroke and cerebral ischemia. Retroviruses are viruses with an enzyme that allows for a reversal of genetic transcription, from RNA to DNA rather than the usual DNA to RNA. The newly transcribed viral DNA is incorporated into the host cell's DNA strand for the production of new RNA. Retroviruses can be converted into therapeutic viral vectors by the removal of certain genes. This vector was successfully used to treat bubble boy disease. Adeno-associated viruses is a parvovirus with a genome of single-stranded DNA. It is non-pathogenic. Most people carry this harmless virus. Most people treated with AAV vectors will not have an immune response. Several clinical trials with AAV are ongoing to treat muscle and eye diseases. Gene expression can be upregulated and downregulated by antagonists, agonists or ligands that affect nuclear cell receptors, a molecule found on the surface of a cell. The receptor receives specific chemical signals from neighboring cells and distant cells that tell it to do something. PPAR receptors function as transcription factors regulating the expression of genes. They play essential roles in the regulation of cellular differentiation, development, and the metabolism of carbohydrates, lipids and proteins. PPAR delta pyroxene proliferatase activated receptor delta modulation activates fat metabolism, reverses metabolic abnormalities and reduces oxidative stress, a factor in aging. PPAR gamma pyroxene proliferatase activate receptor gamma modulation activates slow twitch muscle fibers, increases strength and endurance and induces calorie restriction, an anti-aging therapy. It's also a therapy for muscle-wasting diseases like Duchenne muscular dystrophy. Ribonucleic acid has been used in gene therapies. It is one of the three major macromolecules, along with DNA and proteins, essential for all known forms of life. Like proteins, some RNA molecules play an active role in cells by catalyzing biological reactions, controlling gene expression, or sensing and communicating responses to cellular signals. The human genome, the entirety of an organism's hereditary information encoded in DNA, can be edited to treat disease. RNA interference process cells use to turn down or silence activity of genes by controlling which genes are active and how active they are. There are two types of small RNA molecules, microRNA and small interfering RNA, that are central to RNA interference. They bind to other specific RNA and either increase or decrease their activity by preventing a messenger RNA from producing a protein. MicroRNA binds to complementary sequences on RNA transcripts, resulting in gene silencing. Aberrant expression of miRNA has been implicated in numerous diseases. Small interfering RNA is a class of double-stranded RNA molecules, 20 to 25 nucleotides in length, that interferes with the expression genes. Genome editing identifies precise location of mutated DNA, and targets and repairs genetic defects. Exome sequencing the coding regions of the human genome, or exons, to identify genes associated with genetic disorders. Exons are short, functionally important sequences of DNA that represent regions in genes which are translated into proteins. It is estimated that protein coding regions constitute 85% of disease-causing mutations.